Hello, Morgan. Hi. How are you? Good. All right. Hello, Susan. Hi. All right. So, hope you had any updates, Susan. No, nothing. Welcome. Why don't we get started? I think Susan sent me a paper on cell mechanics. I'll have to go through that. Then we have another paper to go through. Um, this is a nice resource here. This is the Epithelium Mechanics Fan Club. This is something that they do a lot of posting on social media, but they have this organization here where it's early career researchers, and they have a lot of interesting social media threads on epithelia and papers that are coming out. They just have that thread or those threads kind of fed to this website. So, this is a good source of getting new papers that, kind of look interesting and then getting like a tutorial on them. So this is something on positional information. This is something on, well, this is positional information in bits. That's an older paper. This is optimal decoding of cellular identities in a genetic network. And, just there are quite a few papers that I've picked from here that have been really interesting. So that's that. This is the paper on cell-cell junctions I just told you about. This is in tissue barriers. So these are cell-cell junctions, structure and regulation in physiology and pathology. This is a review article, so they go through a bunch of different mechanisms that are involved in these junctions. So let's see what they have to say in the abstract. So in the abstract, they say that epithelial and endothelial cell contacts are established and maintained by several intracellular junction complexes. These structurally and biochemically differentiated regions on the plasma membrane primarily include tight junctions, which are abbreviated TJ, and anchoring junctions, which are not abbreviated, while the adherens junctions, which are AJ, so adherens is AJ, and tight is TJ, and then anchoring junctions are kind of generic. So just keep that in mind. And so the adherens junctions or AJs provide essential adhesive and mechanical properties. TJs hold the cells together and form a near leak-proof intracellular seal by the fusion of adjacent cell membranes. So when you have cell membranes fusing together, you have the seal. That's the tight junctions. So if we think about it as like a seal, and then adherens junctions as like this sort of adhesive, and you wouldn't use an adhesive necessarily to seal things. TJs hold the cells together and form a near leak-proof intracellular seal by the fusion of adjacent cell membranes. Okay. AJs and TJs play essential roles in vascular permeability, obviously, considering their involvement in several key cellular functions such as barrier formation, proliferation, migration, survival, and differentiation. Further research is warranted on the composition and signaling pathways regulating cell-cell junctions to develop novel therapeutics for diseases. The current review article presents our current state of knowledge on various cell-cell junctions, their molecular composition, and mechanisms regulating their expression and function in endothelial and epithelial cells. So this is talking about this, how cells, they're talking about eukaryotic cells here. Cells encompass the fluid matrix, and organelles containing various chemical and biological components are wrapped in a limiting membrane. These are the basic units of structure and function in life. The eukaryotic organisms, with much larger genomes and proteomes, are generally larger and more highly organized than prokaryotic. cells. Last week at the end of the meeting, I did a paper on archaebacterial cells, and there's some archaeobacterial cells, there's a genus of them, that have features that you also see in eukaryotic cells. As um, archaebacteria are more closely related to eukaryotes than prokaryotes. And so they have some of those. I don't know. I don't think they have a common ancestry. They may have a common ancestry, but they have similar features, which means that you may have convergent evolution, and it may be because there's some need in systems that are more complex for these types of more uh, elaborate structures. Cells make up tissues. Tissue comprise organs. Organs form organ systems, and organ systems work collectively to generate an organism and keep it alive. The increased organization within the eukaryotic cells have allowed for the evolution of thousands of different types of dedicated eukaryotic cell types that perform specialized functions such as protection, movement, energy storage, or reproduction. And then they talk about how epithelia and mesenchyme are the most important tissue types in the metazoan body, with epithelium arising first in embryonic development and mesenchyme evolving from it. A switch-off mechanism of differentiation and maintenance of epithelial cells exists. The tissues produce all of the organs of the body through mutual inductions and interactions. The differentiation of blastomeres into an epithelial state is essentially the first coordinated activity an animal embryo undertakes. And so they kind of go through a little bit about development. And then they say, furthermore, cells of the 8-cell embryo in mammals and the blastula stage of animals in general are composed entirely of epithelial cells that are held together by cell-cell junctions. So this is, again, talking about holding the cells together into these sheets that form organs and tissues. And we need these kind of junctions to perform that sort of function, that intercellular function. So they talk about EPCs, which I guess are what they call epithelial cells. That's the abbreviation they're going to use. Epithelial cells from the epithelia, one of the fundamental tissue units of the human body, whereas endothelial cells are ECs, so it's EPCs versus ECs, derived from the mesoderm, or a thin layer of squamous cells that form the innermost structures covering the interior walls of arteries, capillaries, and veins. So they have different mechanical potentials. They're dealing with different types of loads. They're dealing with different types of environments. While the basal side of these cells is attached to the basement membrane, the apical surface confronts the lumen towards spacer blood and EPCs and ECs respectively. So you have, in one case you have flowing blood, on the other hand you have space, which I guess, could be some environment that doesn't have maybe a high flow rate. These cells can collectively form the cellular sheets that act as a physical barrier, playing a protective role against the external microorganisms in animal and human life forms. Cell-cell barriers are established and maintained by the presence of several types of intercellular junctions. Cell-cell junctions themselves help with the attachment and communication between the cells. So we have our cells, and they need some sort of communication. So it's bidirectional. Yeah, that's interesting. Yeah, I know a lot of cells are kind of like that, where they have, you can do this with stem cells too. You can put them under different physical conditions and they differentiate or they don't differentiate. So you can grow stem cells under like on a pattern surface and you can get them to differentiate in one way versus another. And it's just they're adhering to the surface that's either, rough or smooth, and it's introducing physical forces there. And so, yeah. Yeah. Well, it probably, it is a way to kind of keep that regulated, right? You need that cell type. If it differentiates away from that cell type, you have a problem. You have blockages, or you have, yeah, where the surface is not porous. Well, it must be tolerant of something because people with like clogs in their arteries don't like die immediately. They die slowly. But yeah, well, yeah, it's true. Blood vessels, I guess, too. If it doesn't have the proper flow through the blood vessel, you have necrotic. These cells can collectively form the cellular sheets that act as a physical barrier, playing a protective role against the external microorganisms in animals. And so this is just kind of in eukaryotic cells, of course, or in, I guess, eukaryotic epithelia, no, prokaryotic epithelia. Okay. So you get this sort of protection against basically infection, or microorganisms doing something in there. So yeah, that's good. Cell-cell barriers are established and maintained by the presence of several types of intercellular junctions. Cell-cell junctions themselves can help with the attachment and communication between the cells, separation of the intravascular and extravascular compartments, and the maintenance of cell polarity. So this is all, kind of things that you need to get differentiation and development. Remember, those cells have to sort of form this layer to begin with. Then you have to maintain that over time. And you want to maintain, tissue integrity. So if you lose your flow, you don't maintain tissue integrity. But as long as you have this sort of homeostatic set of conditions, you maintain your flow. Experimental evidence has suggested that protein-protein interactions in the cytosol of such transmembrane junctional proteins modulates the extracellular action of the protein, which achieves homophilic or heterophilic binding to extracellular domains of junctional proteins on neighboring cells. So in this way, a controllable intracellular seal is created. So these are, of course, the junctional domains, such as adherens domains, AJs, and tight junctions, TJs. Okay, so that's what they're getting at. So these seals, or these junctions, are very much a product of function, keeping cells together, keeping their identity stable. So this is a nice review article. They kind of go over the different types of cell junctions here. This is figure 1. They have cell-cell junctions. They divide into anchoring junctions, tight junctions, and gap junctions. Then you have here adherens junctions, desmosomes, hemidesmosomes, and focal adhesions. So these are all different types of anchoring junctions. Focal adhesions anchor the cells to some surface. Hemidesmosomes connect to the cytoskeleton by intermediate filaments. Desmosomes connect to the cytoskeleton by intermediate filaments as well, and they're used in different contexts. And then adherens junctions connect to the cytoskeleton by actins. And these are transmembrane proteins such as cadherins, and plaque proteins like catenins. So this is all kind of, adhering to things. And then tight junctions are connected to the cytoskeleton like actins, sort of like adherens junctions. Except they have different transmembrane proteins and different plaque proteins. So the transmembrane proteins here are claudins, and jams. Not sure what jams are, but those are different proteins you'll read about in the literature. Plaque proteins include ZO, cingulin, symplectin, and others. So these are all kind of these tight junctions that form the seal that they were talking about. And then gap junctions, which are these electrical junctions between cells. These are also sort of mediated by transmembrane proteins called connexins. Okay. So one thing about naming proteins, they really do go for like kind of the function. They'll name it something like that. And then where they'll give it an unnecessary acronym. All right. So they're anchoring junctions with proteins present within. I talk about anchoring junctions present near the apical portion of two adjacent cells, just behind the tight junctions. So they say figure 2, adherence junctions, the primary anchoring junctions, protein complexes are formed by transmembrane adhesion proteins called cadherins. Cadherins are particularly important in embryogenesis in the direct anchoring of the stem cells to niche cells, organizing the niche, controlling cell division orientation, regulating signaling pathways, and affecting the mechanics of the cells. So this is kind of the point about cadherins, is that they're important in setting up the tissue, setting up the sheet that will then form this functional surface that we see in the functional biology. So if we're looking at the surface of a vessel or air pathway where there are physical flows, either of air or of liquid, those things have to be built in some way. They have to be built sort of maybe in the absence of that force, but they have to be set up so that when the force comes or the flow comes, then it can respond to that. So these are all important in embryogenesis. Setting up the niche, and then the niche is the thing that is, either built up or broken down by this activity dependence. So if there's a flow, if there's a continual flow of air or liquid, then it maintains its niche. If not, it pops out of its niche, and then you get sometimes you get cancer cells, sometimes you get necrosis, sometimes you get apoptosis, things like that. All right, and then so this just kind of goes through some more details about this. So there are a number of sections here that kind of go into these different components, and they describe them. They also describe a lot of the molecular components. You have, of course, a lot of key proteins and signaling molecules, and so they kind of go into that later. This is figure 2. This is a connection of various tight and anchoring junctions to the cytoskeleton. So if we look at this, we have our endothelial cells, these yellow structures, and they're kind of facing each other. And then you have these junctions here. So you have tight junctions here, where you have actin molecules on either end of the junction. You have then these ZO proteins here, and they serve as anchors on the actin for these claudin molecules, these occludin molecules, and these jam molecules. So these are the ones that we were talking about earlier. And those make the sort of the junction between the cells itself. So they make contact outside the cell body in the intercellular space. And so I assume because this is actin, there's a lot of movement here. It's moving back and forth, or it provides this sort of motor on either end. And then this anchoring junction here, you have an adherens junction and a desmosome. So the adherens junction again is an actin on either side, an alpha catenin, and then on the outside of each one of those, you have a P120 molecule on the junction side of that, and then you have these cadherins that meet in the intracellular space and form the adherens junction. So adhesive properties are made by the cadherin. But again, you have these actin molecules on the other side, on both sides. So there's this potential for like kind of motion or motor thing here. Then the desmosome, and the interesting thing here to note is that it's a cadherin and not an actin on either side. So you have these cadherins, and then you have the desmoplectin molecules on the junction side. Then you have plakoglobin on the junction side. And then you have these other molecules, desmocolin and desmogliin, which form this junction where look like they're sliding back and forth. Yeah, I mean that sounds That sounds interest. That sounds. I don't know if that's what the what the consequence of that is. I guess it's yeah. You get this sort of stretch, and then when you need even more stretch, like extreme cases, you have the desmosomes bifunctionality, where it just gives it the extra. Um, it gives it extra functionality. Okay. Yeah. Yeah. J curve. I think that's what you called it. Yeah, it gives it sort of an elasticity under extreme conditions. It's like if you're pulling apart two things with a vacuum seal. It's like hard to pull them apart, and then once you do, they pop. But it's like that, and then this might mediate that part where they're really trying to be pulled apart. Okay, yeah. So this is, and then this is the two cells, the yellow, and then the extracellular matrix is down here in the pink. And so when it's connecting to the extracellular matrix, you have the hemidesmosome and the focal adhesion. The focal adhesion being mediated by actin, and the hemidesmosome is mediated by cadherins. So that's where we have that. And then we have, of course, our plectin molecule here on the junction side, and then these, what are these? Jam molecules or integrin. Oh, your integrin. So your integrin then bridge that gap across the junction. And then the focal adhesions and the hemidesmosomes mediate like how the cell connects to the substrate, basically. Okay, so this is figure 3, structure of a typical TJ Claudin ECL extracellular loop. So this is just kind of like this. This is the cytoplasm. This is sort of at the membrane where you have these kind of connections here. It says transmembrane proteins. In blue, it's hard to see the text there. And then I guess these are extracellular loops. And these are the NH terminal. This is the PDZ binding motif, the COH terminal. So you have these things inside the cell, and then these loops outside the cell, and then these structures here, just the membrane. So you have these transmembrane proteins that are mediating these junctions. All right. And then this is an example of a knockout where you have, these are the types of CLDNs and their mechanisms. You have two examples of a knockout and two examples of a wild type for barrier forming claudin and pore forming claudin. So the barrier forming claudin in the wild type, you see this, and when you knock out, I guess you're knocking out CLDN1 and CLDN3. So if you have normal function here with barrier forming claudin, and then when you knock that out, you have passage. If you knock out CLDN1, you have skin dehydration, so this layer is forming like skin. So if you can, that the barrier between the cells is forming a seal that is keeping water within the matrix there of the cells. If that's knocked out, you get skin dehydration. If CDLN3 is knocked out, you get sweat leakage. So you just get different ways in which this barrier is or the seal is compromised between the cells in this knockout. So those are two proteins that can be knocked out, and they have different phenotypic defects. Now, pore forming claudin, so it's a barrier forming, so it's keeping things in and keeping things out of this matrix. So you have two cells, there's a barrier, and then that barrier is acting as a seal or a protective layer. Then the pore forming is where you have a pore, and this is where you have selectivity going into that matrix between the cells. So instead of a barrier, you have a selective pore. And then, of course, if you have your knockouts, this ceases to become selective. You can have different phenotypic defects here. So for CDLN10, if you knock that out, you have nephrocalcinosis, and then CDLN15, you have intestinal malabsorption. Basically, defects of this this epithelial layer. So this is kind of showing the effects of what happens when you knock out different proteins and showing that effect on the phenotype. This is figure 5. This is regulation of adherens and tight junctions by SRC and AKT. So this is, again, a pretty busy diagram where you have different things going on here. You have these junctions. You have a tight junction, adherens junctions. You have here barrier leakage. And so you can have leakage between these junctions if they're not properly formed. You have, these are basically claudin disruptions, and those result from downregulation of claudin. You also have V-cadherin internalization, which is again a product of the molecular milieu in one cell or another. So you can have underproduction of different factors, and they can result in this barrier leakage. You also have different receptors on the surface, and they lead to different things going on here. And it looks like here that you have this circuit, this molecular circuit. You have FOXO1/3A, which is important in claudin regulation, and you have SRC and MDA, which are important in V-cadherin phosphorylation, which leads to V-cadherin function. Okay, so I think that's it for this review. They have some concluding thoughts here. So considering their involvement in several key functions of the cells, including barrier regulation, proliferation, migration, survival, and differentiation, cell-cell junctions need to be studied in more detail. Their aberrant expressions are not only a cause of several diseases, but also reliable markers for diagnosing these illnesses. The current review portrays the reciprocal regulation of AKT and SRC and the maintenance of adherens junctions and tight junctions. So that last figure was about like how AKT and SRC are modulated by these molecular circuits, they're modulated by receptors, they're modulated by the need for a junction between two cells. However, these mechanisms have been studied in isolation, and therefore it is not clear how distinct signaling mechanisms crosstalk with one another. So we haven't even talked about crosstalk yet. We've just been looking at these idealized models of function. Much work remains to define the contribution of individual signaling molecules, which can then be targeted by pharmacological or genetic modulation to treat VP-related diseases. Hence, further research is warranted on the signaling pathways involved in the regulation of cell-cell junctions. Okay, so that was that paper. Thank you, Susan, for bringing that up. We had a lot of classification, a lot of useful information on junctions, and they showed some experimental data, basically just giving a quick sketch of how that's all regulated and how you can end up with different pathologies and dysfunctions. Okay, yeah, 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 yeah. All right. So yeah, that's that paper. Why don't we talk about this one? This is interesting. This is called cell supracellular structural relations solve the French flag problem without graded molecular control. So this is by Amy Shire and Alan Rodriguez and colleagues. We talked about Amy Shire and Alan Rodriguez as having, they're doing a lot of work on morphogenesis and systems and introducing some mathematical models in there. So it's pretty interesting work. They're from the Laboratory of Morphogenesis at the Rockefeller University. So this is, what they're looking at here is they're looking at the French flag problem. So the French flag problem is how you get sort of a French flag from sort of a random array of cells. And so what that means is in morphogenesis, usually we're interested in pattern formation. And a lot of times in development, that manifests itself in striping. So if you look at the Drosophila model organism, you look at the models that they have in Drosophila in development, you have this striping that occurs, where the segmentation is another way to think about it. And that results from these gradients that intersect. These gradients are morphogens. And they're spatially restricted so that those boundaries are very sharp, and you get these stripes or these segments that are very distinct. And so if you think about the French flag, it's a series of stripes of different colors. And so that's what they mean by the French flag problem. You have these stripes of different colors, and how do you get the French flag from basically random order, or disorder? So this is a self-organization problem, and we're trying to understand how spatial patterns of cell state emerge. So these stripes will have cells of a distinct state, and they'll be separate from another stripe where you have cells of a different state. So the abstract reads, the molecular characterization of tissue organization through spatial omics. And so they're characterizing things through this spatial transcriptomics, or these spatial proteomic approaches, where you get local information, like sometimes it's single cell information, and it tells you something about that process because different cells represent different points in space, or sometimes you can sample like a tissue where you can, or like an embryo where you can isolate different transcriptional profiles at different parts of space. And then you can answer questions like this. So we use spatial omics. We can take a new kind of look at the French flag problem. So Wolpert's French flag problem was about how spatial patterns of cell state emerge. Here we address the skeletal pattern of adjacent cartilage and soft tissue. So this is actually in a complex tissue where they're looking at cartilage and soft tissue within a skeleton. We find that cellular and supracellular structures are co-constitutive and result in cell, extracellular matrix, and cell-cell-based structures that canalize cartilage and soft tissue fate, respectively. So this means that you have this canalization process, so things get buffered or channeled into certain fates based on their context. A bistability in these fate-setting structures establishes pattern length scale and is sufficient to break symmetry in the mesenchyme. However, an adjacent epithelium, through signals like Wnt, can locally seed the bistability to ensure the epithelial adjacency of soft tissue. So basically you have different, you have cells, and they're, kind of don't know what fate they need to be in, and based on their sort of location in the tissue, they are restricted to certain fates, and that sets up these boundaries between different types of tissue, and those boundaries are very important for function. So you have an epithelial on one side of the boundary, and then soft tissue on the other side of the boundary. And that you have to get that sort of adjacency correct. So you have to have the right cells on each side of that boundary. You also have to have, you can't have cells in between that aren't quite sure of their fate. because it has to be a hard boundary. You have to have an epithelial sheet and then soft tissue right adjacent to it. For functional reasons, that's very important. So it's important to have this sort of mechanism that allows for this boundary to be set up and to be sort of error-free. And so a bistability in these fate-setting structure, okay, we did that. However, an adjacent epithelium through signals like Wnt can locally seed the bistability. We did that. Rather than acting in a graded manner to provide positional information, Wnt affects cell fate by promoting a supracellular structure. So Wnt is basically creating the supracellular structure and controlling cell fate in that way. Together, these results support a solution to the French flag problem, where fate pattern originates through structural relations between cell and supracellular levels. So patterning actually is about these structural relations. So you have cells and they're acting on their own. You have these supracellular mechanisms acting on the cells, and you get this organization. So that all sounds good, but let's look at the paper a little bit more. So the French flag model was introduced by Louis Wolpert, and that's sort of the classical model for this type of patterning in different structures. So again, you get this, you think of the French flag, it's 3 stripes. Each stripe has a sharp boundary, and this is the thing we need in tissues to distinguish between different types of tissue within the structure and perhaps different types of function. So they describe the French flag model as where differences in signaling molecule concentrations generate subcellular positional information across the tissue. So you're actually interested in subcellular positional information being sort of this, where is the cell? What is it supposed to be at this location? And there are a number of schools of thought on this, that positional information is key to this sort of, structure building. And then the question is, well, how does it know exactly what its position is? And so this is done through coordinated molecular activity. At least that's how a lot of people have thought of this. So you get these differences in signaling molecule concentrations, where the cell can sense the concentration of different molecules and know its location in this gradient. And then, of course, there's a templating to corresponding structures. So if you know your position, then there's this sort of template. You kind of understand where you are in the template, and then that's the fate that you take on. And it's all that all sounds very cognitive, but this is all sort of a cell sensing its gradient and then knowing what the fate is supposed to be. It might be done through thresholding or some other mechanism. And so this works out pretty error-free in development. Here we identify a novel mechanism for the organization of compartments within a tissue that does not rely on graded molecular control. So the French flag model is based on this graded molecular control concept. They're finding, in this case, a novel mechanism which does not rely on this. Instead, we find that fate patterns originate through structural relations between the cell and supracellular levels of organization. So in complex systems like organs, the structure that form and the potential for such structures to reciprocally adapt in response to relations within and across levels, enabling organizing processes that are unique to biology. So basically, we have the cell, which is at this sort of lower level of organization, the supracellular, which is this higher level of organization. And I think what they're doing is they're thinking about this in terms of like complex adaptive systems. So you have these lower levels, and they inform the structure of the higher level, but the higher level then constrains the lower level. And there's this reciprocal information or this reciprocal loop that forms. So there's information guiding the supracellular level, and these constraints from the supracellular level constraining what the cell can do. And you see this again and again in biology with these different levels of organization. So, how do you form a complex structure that is repeatable and error-free, or at least it's, repeatable in the sense of being very self-similar? Well, you get these different types of regulatory signals. You basically it's basically self-organization, and so that's kind of what they're getting at here. And so these reciprocal relations enable organizing processes that are unique to biology. Such adaptive and reciprocally causal relationships are specific to living systems because they stem from evolutionary processes fueled by adaptation and therefore may not be simply derivable from physical or chemical first principles. So again, this is saying that, this isn't like chemically deterministic or physically deterministic. In other words, it isn't guided by physical constraints alone. It isn't guided by chemicals signaling matrices alone. There are this need for adaptation, in maybe across species or across orders. You have a lot of conserved molecules. You have a lot of conserved physical processes. So if you see these changes across evolution, then how do you get these changes in the same system? Do you get this through just merely through chemical changes, or do you get this? Maybe they're physical changes, but those physical changes probably aren't that great. So how do you get these, evolutionary changes? And the answer, of course, is through these kinds of self-organized or complex adaptive systems. So they give this example. Unlike living systems, water molecules do not fundamentally change within a snowflake as it forms, nor do Lego blocks transform as they build, or as they are incorporated into a structure. However, cells can adapt in response to the structures they build. Along these lines, while physical and chemical principles and tools are fundamental for studying living systems, as exemplified by the power of mechanochemical frameworks, additional principles may be necessary to fully capture certain aspects of living systems. So this part here, cells can adapt in response to the structures they build. So as the structure is being built, that structure then is continually kind of regulated by the structure that's there. So, it's kind of like if you think about maybe like a termite mound or an ant nest. The organisms are interacting, they're playing their role in building the structure, and depending on the sort of the state of the structure, continually building onto the structure is going to be affected by the sort of the current state. So the future state affects the current state. The same thing with a lot of other complex adaptive systems where you have an initial condition and you build from that initial condition in a certain direction. And then depending on what those current states are, they inform future states. So if you have maybe the same starting point and you go in different directions, you can end up with radically different types of structures. But the principles behind the building of those structures are the same. The chemical principles and the physical principles are basically the same. It's just that you're going in a different direction. Um, and so that's kind of what they're getting at. And they don't propose any principles that sort of govern that. It's, the best analogy, like I said, I can make is sort of these emergent structures that are built by social insects. I mean, that's the best way to kind of visualize this. And then you transfer it to sort of cell biology. And, you can kind of think of, the cells as the individual agents, and then they're building something greater than some of their, like a epithelial sheet through kind of their interactions and forming these structures and how the structures are put together. So that's kind of where we are with that. So a framing of tissues through the concepts of ecology, in particular, focus on the mutual and often simultaneous relations of structures across scales, and the associated organization may be a powerful complement to current mechanochemical frameworks. So this is again where you have these sort of ecological models that are basically complex adaptive systems. So in ecological models, you have trophic levels and you have interactions between trophic levels, and you have this very, some people call it fragile, but it's actually also quite robust set of relationships that can persist. And there are things where you can perturb it in certain ways and the thing will collapse, or perturb it in other ways and the thing remains stable. And this is not necessarily predictable, but it does provide um, this sort of structure that is particularly suited for living systems which change constantly. Much in the way that ecologists must be attuned to the adaptive behavior that occurs through reciprocal relations within and across the hierarchy of levels of organization, there is an increasing need to examine their ecology within tissues and organs. So a lot of times in developmental biology, we use terms like niche in, cell biology in general. And niche is something that comes from ecology, and it just means like sort of a local optimum for some organism that finds its niche in an ecosystem. And you have cell types that have a niche. So there are certain conditions under which the cell type will thrive. And so this is kind of this ecological metaphor. What they're saying is that you need to think about the ecological metaphor more broadly and bring this into like thinking about tissues and organogenesis. While the aspects of living systems that set them apart from inorganic systems have been theoretically considered, empirical examples are still limited. Without clear and concrete illustrations of the particular nature of the structures and organizing processes across levels and organs, our ability to grasp causality in health and disease, and their successfully intervene may be limited. So that's kind of what they're talking about here. Let's go into maybe some of the figures. Okay, so this is figure 1. You have this mesenchyme and ectoderm. You have this development over time, and you have these stains where you have SOX9 and twist, and then this is merged. You have this other image here, which is where you have the expression over time. So you have the ectoderm here, then you have where there's differentiation, and you have SOX9 changing its profile with respect to twist, and then the DAPI stain here. This is spatial transcriptomics, where you have this profile by radius, so in microns. So you see that there's this, as you go outward, you have this drop off and twist, this drop off and DAPI, as you might imagine, because the cell body is getting to the edge of the cell body, but then SOX9 increases here. So this is where you have SOX9 twist GFP stain, so it's co-stained, and you can see that here. And then you have different types of tissue, SOX9 for cartilage, soft tissue. So for SOX9, you have in cartilage, you have more of that and less of that in soft tissue. For twist, you have less twist in cartilage, but a lot more in soft tissue. So that's showing the spatial aspect of expression. You have DAPI and cadherin. You have this comparison in cartilage and soft tissue. Again, you have differences in terms of the tissue. So they're characterizing the different tissues and gene expression across those tissues. And the idea is that there's this hard boundary between cartilage and soft tissue, and this is spatially distinct. So we have this radius from, a single point, and we can see these differences in gene expression, and then those are very distinct between cell types. Figure 2 is where you have the supracellular level impulse for cartilage fate activation. So again, we have some molecular characterization of this. And then at the bottom, they give the interpretation, which is where we have Lin mesenchyme, which is naive. We have extracellular matrix, which is knocked out. We have cell extracellular matrix calcium signaling, where we get this difference in cell types. And then we have this turns into the cartilage progenitor. And then there's this top-down causation, which leads to this SOX9 positive cartilage cell. We have then these other cells from the Lim mesenchyme that go down this other path, where when we block off this ECM, we end up with cells that are undifferentiated that are SOX9 negative. So that's basically what they're showing. SOX9 is sort of the driver of this process. And given the ECM, the presence of an extracellular matrix, and this calcium ion signaling, you either get these very specific cells, these cartilage cells with the SOX9 marker, or you don't. So that's what they mean by top-down causation, that the extracellular matrix is playing a role here in shaping which cells are going to be which thing. In this case, it's shaping which cells are going to be cartilaginous, and you get all these cells that are cartilaginous. You start to get this early with, some cells differentiating and some cells not, but then all of the cells at the end of this process are differentiated and have a strong positive signal. This is figure 3. Wnt activation of soft tissue fate is mediated by cell-cell interactions, and this just shows more characterization. We look at the cartoon again. They show a little bit more about this. So we have Lin mesenchyme, which is naive. We have this transition to cartilaginous cell type with SOX9 positive marker. But then if we go to this intermediate stage and we look at the cell-cell interactions and we look at Wnt signaling, we have the soft tissue progenitor. So we take some of these cells. We have these cell-cell interactions. We introduce Wnt. We have soft tissue progenitor cells that are twist positive instead of cartilaginous progenitors, which are SOX9 positive. So you see that they have this different shape and they have this different thing that's being expressed. So you see that there's this cell that's in isolation. If you add or if you overexpress Wnt in that single cell, it's twist negative. But if you have a bunch of cells in a population, they're all together, they're adhering to one another, and you upregulate Wnt, then you get these twist positive cells. And so that's the difference, is that they're in this extracellular matrix. They're all kind of joined by these junctions, and that's the sort of the higher layer that's constraining what these cells can become. So again, if you have the cell in isolation, it doesn't respond to Wnt overexpression. It has to be in this cell-cell interactions to get the twist positive. So again, this is what's happening with, in a cell population. You have these Lim mesenchyme, which are undifferentiated, and depending on their local environment, depending on how the cells are interacting with one another, they form these different cell types. And you can verify that they're of that cell type by their molecular signature. So this is top-down causation. Figure 4 is cartilaginous and soft tissue domains can arise in the absence of a spatially located epithelial signal. So if we look at the bottom here, well, first of all, we can look at DAPI and encadherin for task and the control, and we can see that there's this distance from ectoderm in microns. You have this normalized mean integrated DAPI intensity, and it decreases. But they show their result here, their model. So you have cellular motion, where you have pressure, stress, and traction, and you have gradients for all three of these. And then you have this ECM transport gradient, which leads to extracellular matrix synthesis. So you have transport in the extracellular matrix, which is a gradient. You end up with synthesis, and this is a product of pressure, stress, and traction at equilibrium. So, there are all these physical mechanisms going into cellular motion in the extracellular matrix. You also have extracellular matrix mechanics. So you have viscosity mu, which is the product of the interfacial tension gradient, the cell adhesion, change in cell adhesion, and then what we call haptotaxis, or movements based on touch or pressure. And then, of course, we have repulsion. So we have these physical signals, these physical cues. We have changes in adhesion. And we have this gradient of interfacial tension, and that leads to viscosity. And then we have cell advection and cell diffusion described by this differential equation. And we have these gradients of cell advection and cell diffusion that are additive. And then feedback mechanisms, we have the cell extracellular matrix engagement. You have this differential equation that describes these gradients here, and then cell-cell engagement. So the cell extracellular matrix engagement is a single cell in response to its extracellular matrix. The second term is cell-cell engagement, which is where the cells are interacting directly and independently of the extracellular matrix. And in this differential equation, we have mixing, which is a gradient, advection, which is a gradient, and these bistable kinetics, which are part of both terms. If we look at this here, we have the difference. So we have for extracellular matrix density and cell density, we have these different outcomes. So as we increase the T parameter from 0.83 to 1.5, we get these differences in, I guess, this metric here, which is more, I guess, more activity, but it looks different. And so, yeah, you see that like extracellular matrix density and cell density give you differences in this. As you increase this T parameter, this increases its activity. I'm not really sure what this is exactly showing, but it's in relation to this model. So cell and ECM densities in control micromass simulation at 3 time points, 0.83, 1.16, and 1.5. And so that's kind of what they're doing here. This is showing Wnt concentration, extracellular matrix density, and cell density. And these are just showing the radial distance and showing this cell density and ECM density, comparing the two. So with Wnt concentrations, the cell density is this aqua line here, and ECM density is this gray line, and then Wnt is this purple line. So you see Wnt decreasing with radial distance, cell density decreasing with radial distance but remaining above 0, and then ECM density increasing with distance after a radial distance of 2.5. So you see the ECM density rising where cell density is decreasing. That makes sense. And then Wnt is decreasing quickly, even before cell density drops off. And then the simulation, you see a similar thing and a similar thing here. So all of these just show this relationship. And then finally, we can show this cartoon here where we have fibronectin around this structure here. Cell density twist is in the inside of the structure of fibronectin, and then the ectoderm is within that. So we have these three conditions. We have low cell ECM engagement, control, and high cell ECM engagement. So in the control, we have this pressure from fibronectin inside towards the ectoderm, and this is putting pressure on cell density. In the low cell extracellular matrix engagement, we have ectoderm cell density twist, which diffuses over distance and gets weaker as you get towards the fibronectin layer, which is then more diffuse. In this case here, which is high cell extracellular matrix engagement, you get this a lot more fibronectin. It is migrating out from this ring and it's moving outward. And then you have the cell density twist, which is dropping off from its central source and becoming weaker as we reach the fibronectin and then the ectoderm in the middle. So they can model this for kind of like average levels of extracellular matrix engagement, low cell extracellular matrix engagement, and high cell extracellular matrix engagement. And it just shows you the results of that. So the extracellular matrix is playing an important organizing role here. Okay, so I think that's all for that paper. Any comments or questions? I think it fits into the previous paper in that it shows that these junctions play a role in extracellular matrix and these relations between cells. And so you can imagine that there's this interaction between extracellular matrix, which is the stuff outside of these cell layers. The junctions are playing roles in regulating what maybe goes down into the substrate, what goes down beneath the cell layer, and what stays above it, and how those things are shaped and differentiated. All right. Well, that sounds great. Yeah, so I guess that's all for today. I'll keep thinking about this stuff and send papers as appropriate, I guess. All right. Thank you. See you next week. So I wanted to finish up with this paper. This is a newer paper that came out on the archive. It has Renal Lambio and Elaine Garelli does this stuff with physics, biophysics, and physical aspects of the brain and things like that. And Renaud Lambio does a lot of network science stuff. So these are good people for this paper because this paper is titled From Reductionism to Realism, Holistic Mathematical Modeling for Complex Biological Systems. So they're going to get into modeling and some of the aspects of modeling, philosophical aspects, technical aspects that we need to think about when we apply a model to a system. So the abstract reads, at its core, the physics paradigm adopts a reductionist approach, aiming to understand fundamental phenomena by decomposing them into simple, elementary processes. While the strategy has been tremendously successful in physics, it has often fallen short in addressing fundamental questions in the biological sciences. This arises from the inherent complexity of biological systems, characterized by heterogeneity, polyfunctionality, and interactions across spatiotemporal scales. So in the Shire and Rodriguez paper, we talked about how they're using this sort of complex adaptive systems approach where you have top-down constraints and bottom-up information to describe, something like pattern formation. In this case, we're dealing with systems that are nonlinear, but also have these emergent properties where you can't break all of the effects down, or you can't explain them in terms of using reductionist techniques, and you can't explain them in terms of fundamental units. So this is kind of some things like heterogeneity and polyfunctionality and interactions are all features of these kind of emergent systems. Nevertheless, the traditional framework of complex systems modeling falls short, as its emphasis on broad theoretical principles has often failed to produce predictive, empirically grounded insight. So this is kind of a criticism of complex systems research where, we're kind of focusing on these broad theoretical principles. We haven't had really good empirically grounded insights. Some of this is due to a lot of these models being very generic. So we can apply, say, like a complex adaptive systems model or a network science model or some other model of emergence to many different types of systems. And they describe the properties of those systems. We can identify, say, for example, emergent properties in things like chemical systems or social insect communities or developmental biology. But, finding the real strong predictive mechanisms that we can find with reductionist techniques using those kind of general approaches, there's a gap there. Something's lacking. And some of that is because we're using these models that are sort of focus on the overarching dynamics. So it's easy to sort of build a very tightly predictive model of a specific system. You could apply sort of a statistical test, or you could apply a specific hypothesis to one limited system, and you can have good predictions because you're focusing on sort of the properties in front of you. But in this case, we have this metaphor, and we say there's this broad principle that we want to apply to the system, and developing a sort of a quantitative technique for that is hard because you have to operationalize it across a wide range of systems, and not all of those systems are equivalent, but they may indeed exhibit these kinds of broad principles. So it's a little harder to get these kind of empirically grounded insights from these when you cast your net so widely and you're using these metaphors instead of actual sort of operational tests. That doesn't mean that it's not worthwhile. It's just a different kettle of fish, as they say. To advance towards actionable mathematical models in biology, we argue, using neuroscience as a case study, that it is necessary to move beyond reductionist approaches and instead embrace the complexity of biological systems, leveraging the growing availability of high-resolution data and advances in high-performance computing. We advocate for a holistic mathematical modeling paradigm that harnesses rich representational structures such as annotated multi-layer networks, so the network aspect, employs agent-based models and simulation-based approaches, and focuses on the inverse problem of inferring system dynamics from observations. We emphasize that this approach is fully compatible with the search for fundamental biophysical principles and highlight the potential it holds to drive progress in mathematical biology over the next 2 decades. So this kind of gets into what we want to do. So they kind of highlight some of these models that are sort of reductionist versus some of the broader principles of complexity models. So they use an example from neurobiology. We start from one of the most important breakthroughs in mathematical biology, the Hodgkin-Huxley model for electrophysiological excitability. So some species of squid possess a so-called giant axon up to 1.5 millimeters in diameter that controls water propulsion. By isolating this large axon, Hodgkin and Huxley were able to insert voltage clamp electrodes in very extracellular ionic concentrations to develop a complete set of ordinary differential equations that describe the propagation of electrical signals through a neuronal axon, providing the first complete description of the action potential, the basis of all neuronal communication. So this is an example of reductionism in modeling, and this assumes that a complex phenomena can be described in terms of a series of simpler, more fundamental processes. So for the Hodgkin-Huxley model, the key assumption was that neuronal activity could be modeled as an electrical circuit, with each component of the cell being represented as an electrical element. This in turn abstracts away other subcellular and biochemical processes. And so this has been very successful across the sciences. We know that many physical systems can be organized onto a spectrum spanning from the regular to the random. And we'll get to figure 1 in a minute, but they illustrate that in figure 1. It is well appreciated that the reductionist approach appears to be the most effective at the extrema of the spectrum, where interactions present certain types of symmetries. At one end of the spectrum, for systems characterized by regular symmetric organization, such as atoms in a metal or crystal individual elements, such as atoms in a metal or crystal, individual elements can be abstracted away through continuum assumptions. So we can use continuum mechanics or partial differential equations to sort of represent the broad population of atoms instead of modeling individual atoms. Then at the other end of the spectrum, in order to describe random systems composed of enormous numbers of noisy interacting components, we can use statistical mechanics. So in this case, we assume that interacting elements are identical. And we don't need to describe the behavior of each element, but we can describe sort of the mean behavior of those elements. And so the dynamics are the things we're interested in, the ensemble. And so this is what, and then we can use mean field approximations to characterize an ensemble, and that describes the state of the system without problem or without incident because everything is homogeneous. And so between these two extremes lie systems that, for lack of a better definition, are best described as complex. So these are where we have topologically irregular interactions, heterogeneous elements, polyfunctional processes, and evolve over multiple time and length scales. So basically anything that is biology. So you can see the problem that in physics, you have these cases where you can't, or at least we have a good characterization of these homogeneous systems, of these systems where we can drill down to like individual elements. And then even with their Hodgkin-Huxley model, we're dealing maybe with a biological system where there's a lot of complexity. We can remove a lot of that complexity and get down to sort of the basic physics of the process. Are we missing something with Hodgkin-Huxley? I mean, we may be missing things. I mean, Hodgkin-Huxley is predictive. It gives you good answers. It gives you, you can characterize a lot of things with Hodgkin-Huxley, but it doesn't describe the biology in the same way that maybe a complex systems model would. But the complex systems model might not be able to give you the kind of predictive power that you would get out of the Hodgkin-Huxley model. All right, so they talk about this distinction between, or actually it's a distinction between three things. You have the purely reductionist model, which gives you these sort of highly abstracted models, which are very atomistic, and they give you maybe really good predictive power. You get these systems that are sort of averaged or continuum systems, which still have predictive power, but rely on these sort of homogeneous elements. So you have, populations of atoms or populations of cells that are all homogeneous, so we can make certain assumptions about their constituency, and we can look at the ensemble. We can look at the average, the mean behavior. But then we get to systems like biology, and you can use those first two models in biology. But, for a complete description of the biology, you need to turn to a third system, which are these complex dynamics, these complex systems. And we saw an example in the last paper, in the Shire and Rodriguez paper, where they were looking at the sort of complex adaptive systems or type of organization. And, that was, I don't know how predictive it was, but it definitely was demonstrable. You could see the differences between cell types. You could see the influence of the extracellular matrix, but we couldn't necessarily model it directly. I mean, they were doing some modeling, but it wasn't exactly giving us a predictable outcome. It was just kind of demonstrating what this process looked like under different parameter values. So this is kind of where they're going with this. They have these three categories, and then they are thinking about how those relate to predictability and how they relate to sort of fidelity in description. So this is the spectrum of interaction models and mathematical models. So basically what they're trying to get at here is that you have this regular regime at the left, this random regime on the right, and this complex regime in the middle. So the regular regime is defined by this lattice. Everything is homogeneous. If I take one element, it can describe any other element. And the connections are uniform, so even the connectivity is sort of the same across the lattice. And then random, of course, is the opposite of that, which is where you have random positions. The connections in the lattice are of different lengths, and the cells are all heterogeneous, meaning they have different behaviors. But it's not something we can actually predict because it's random, and we can't really devise a statistical test to deviate random from random. Usually, when we do a statistical test, we look at the random case versus sort of the ordered case, the sort of the control versus the treatment, the control being sort of a random effect or no effect, and then the treatment being the thing with the effect, the order in it. The experimental manipulation has the order that we're looking for. And so this is regular, random, and then complex sits in the middle. So complex has features of the random. It's heterogeneous. It's irregular, but it's also regular. There's some regularities there. And so that's what we're looking for, is this complex regime. We can't necessarily use a reductionist set of techniques. We may be able to use some continuum techniques or some ensemble techniques to describe the complex, but it doesn't fully describe it. And then to make things worse, you have this regime between complex and random, which is, term, for lack of a better term, the edge of chaos. And so chaos being sort of the random, the edge of chaos being at the edge of the complex, shifting towards the random. And it's sort of systems that exist very, very complex regimes, very close to being random, but aren't random because they actually do generate predictable behaviors and other things like that. So there's some order there. And so, but then, there's this continuous struggle to measure these things and to characterize them completely. And that's what they're kind of arguing in this paper, that this is a very hard thing to do. And they kind of give some examples of how you might do this. So one example of a higher order representation that they give is an annotated network. So, if you think about a network, it's a simple structure. It's a graph. It's G equals VE, which is the vertex set and the edge set combined to form a graph. Annotated networks can provide nodal metadata in the form of an additional vector of features D, and D sub I included in RD, that is attached to each vertex or to each edge. So to V and to E, you would attach this set RD, which would give you this additional information in G. So it gives you, so it's basically, they say it's nodal metadata, but it's really just like introducing heterogeneity into this graph structure. And so in particular, this extension allows us to model the heterogeneity of individual elements, adding a crucial layer of additional expressiveness to our mathematical models. Such an approach has proven useful in neuroscience in the form of biologically annotated connectomes, which are human brain networks obtained from structural imaging, where each node or region is imbued with additional information, such as cell type density, gene transcription, receptor density, and myelination. And it's worth saying that this is, kind of the standard in systems biology, where you have this network of molecules or the network of cells sometimes, where you have, annotations available, and you attach them to the nodes to describe sort of the things that are being expressed in the cell, or the expression values for different molecules or whatever. So it gives you this additional layer of information upon which to act and have this sort of, differentiation within the network, or, whatever structure you're using. And it gives you this, gives more fidelity to your biological model. Open source software such as neural maps facilitate the integration of this multimodal data into richer structural representations. Such heterogeneity has been used to develop biophysical models of brain dynamics that incorporate regional heterogeneity through variations in gene expression or synaptic strength for added biological complexity and closer agreement with observed neural dynamics. So this just shows these richer representations. You have networks on the left, hypergraphs, which we've talked about in the group, which are nodes within nodes, and you have hyper edges within edges, or edges within hyper edges. You have simplical complexes, which are these networks that form shapes. So this forms a triangle, and you can characterize the simplices as different shapes that are formed by connections between nodes. So you have triangles, you have rectangles, you have diamonds, things like that. Those are not, there's a type of network analysis called motif analysis. This is not quite that similar, but it's basically describing your network connectivity in terms of the geometry, in terms of the topology. Then you have temporal, which we did temporal graphs this summer. It was Jayadratha's work. And this is where the network evolves, the connectivity evolves over time. Multi-layer networks where you have different layers. They could be these different layers of organization and biology, from organs to tissues to cells to molecular networks. And they're all connected because those networks are all related, so you have these multi-layer networks. And then annotated networks where each node has different values over time or different properties which evolve independently. So, in network science, it's interesting because there's this movement towards thinking about nodes as not just these single units, but thinking about the internal contents of the nodes, because they often represent many different things. They can represent, people, places, things, objects, whatever. And the principle behind it is that those nodes, no matter what they are, then connect to other like units, and they form these patterns that you can then, through statistics, through ensemble statistics, and through other types of analysis, you can understand that system better. Is that predictive? It's somewhat predictive, except that the nodes have very different properties. They have very different levels of complexity and everything else. And so there's been this movement in network science to kind of think about how these networks are structured, and to think about different types of integrating different types of nodes and integrating information within the nodes, and so on and so forth, hence all of these different variations in network representations. So they make the point here, in turn, the mainstream within these fields may replace reductionist abstractions with richer representations that are capable of incorporating the heterogeneity seen in experiments. To achieve this goal, integration with multimodal data is crucial, but it is worth noting that the scaling challenges prove limiting to this point. So given a system with n elements, the number of pairwise interactions scales at n squared. If we introduce higher order or multi-layer interactions, the parameter space scales at an even faster rate, n to the power of b, roughly. So that's actually like an exponential sort of rate of expansion of our model. As a result, such over-parameterized models are plagued by both the lack of sufficient multimodal data and the inference algorithms needed to fit them. However, as we progress into the age of big data, modern experimental techniques are able to record multimodal data on an unprecedented scale, particularly in biological systems. Moreover, novel techniques in computational statistics and machine learning are able to perform parameter inference for such complex models using observed data. And so, unlike purely statistical models such as machine learning models, these systems have parameters that remain both physically constrained and meaningful. So, if we talk about like a 6 trillion parameter model in deep learning, the parameters they're talking about are very different than the parameters we're talking about in these kind of network models, because we have parameters that are physically constrained and meaningful in the world, or they have different, they describe different properties of the graph itself. This ensures the interpretability of complex dynamical models and allays the usual concern about von Neumann's elephant and its wiggly trunk. So that is in reference to this idea that von Neumann had about being able to predict, or having, like, if you give me 5 parameters, I can model an elephant, and with 6 parameters I can wiggle its trunk, meaning that you can describe sort of a 2-dimensional drawing of an elephant using a polynomial, a 5th order polynomial. Each order of that polynomial is a parameter, and then with that 6th parameter you can model the movement of the trunk part of that diagram. And so, if you just kind of model something with a high order polynomial, you can describe any shape you want. And so the sort of the drawback of these high dimensional models is that you're just using so many parameters that you're just kind of reproducing the the object with a description of shape, maybe instead of the actual thing that you're trying to predict. So that's what they're getting at with that. With accurate calibration on experimental data and efficient simulation on high-performance computers, detailed heterogeneous and elaborate models of complex systems can make the leap from generic and descriptive to mechanistic and predictive. So then they get into different types of models. This is interesting. This is spatiotemporal scales in Alzheimer's disease. This is where we have a very complex disease with different, presenting different symptoms, having different mechanisms, and at different time scales. So we have this, we have space in meters and time scale in from milliseconds to years. And so we see that, like, depending on what we're talking about, we have very different spatial and temporal scales. So protein-protein interactions occur in a very small space at a very short time scale. Cell death occurs at a longer time scale at a little bit larger spatial scale. We have then dementia, which is maybe like 10 years time scale, but in a very large space of the brain. We're talking about like a meter of the brain. So we're talking about the cortical sheet mainly, and we're talking about how much of the cortical sheet is affected by this one process. How long does it take to unfold? So you see that, there are these sort of manifestations of the disease that are at this large space and increasingly larger time scale, but these molecular characterizations that are at smaller time scales and smaller spaces. And so we can use simulation-based modeling, we can do agent-based modeling, personalized biomedical recordings, using an in silico model to look at those recordings, and then building what they call digital twins of the process, which are, so you have two things here. You have data informing sort of high fidelity models, and then agent-based models, which give you this sort of possibility space. So you can use the digital twins and then simulate them in the agent-based model to give us the possibilities within our system. And so finally, I'd like to make a comment about some of the sort of the model zoo they throw at you later in the paper. There are different mathematical approaches which people use for different systems. They're kind of getting at how to apply mathematical models in a way that is suitable to the problem you're dealing with. So not every mathematical model fits every problem. You have different types of time series modeling. You can use differential equations. You also have hidden Markov models, which are good for finding latent variables and states. You have other types of dimensionality reduction or other models to sort of understand higher dimensional spaces and reduce them to lower dimensional spaces. And so they talk about all of that here, and they talk about that in terms of the representation. So I guess I'll address this aspect too, the fundamental physics for biology. We have argued that theoretical biology should turn towards complex empirical models with predictive capabilities, even if these models occasionally complicate traditional mathematical analyses. This shift does not negate the importance of identifying fundamental theoretical principles that govern biological systems, but instead act to support that. So we really need to think about sort of developing a theoretical physics for living systems. So we don't want to just port things from theoretical physics to biology. We want to develop a theoretical physics of biology. And this is based in non-equilibrium thermodynamics and, some of the things that brings us.